start. I'm Gary Leswing. I'm extension educator in Nemaha County and kind of cover like the six counties in the corner of the state. And I've been doing the weed thing since we started this. So we're going to go over that. And I'll let Megan and Nathan and Michael introduce themselves.
Hey, I'm Megan Taylor. I'm in Platte, Boone, and Yates counties, and I'm going to do the corn development and some of the general scouting.
Nathan Mueller. I cover Saline, Jefferson, and Gage County, but formerly Dodge, Washington, and I know Brooke well. She was in our 4-H club, and so I'm excited to see them joining. And I have been covering the soybean growth stage and development, I think, for 5 years now.
Michael Snoller. I work out of Clay, Fillmore, Knuckles, and Thayer. I'll be working on the soybean staging, maturity, and some general scouting around soybeans.
Very good. So, Gary, do you want to go first, or?
Sure. Can you
Do I need to make you?
let me share my screen?
Yes. There you go.
Oh, I know what happened to it.
Or if Megan's ready, or Nathan, I wasn't sure who's.
I was planning just to review a few things in the 2019 corn and soybean pocket guide, just some highlights or some things to look at. And I can do that whenever.
Yeah, no, that's fine. And they don't have them quite yet. I was kind of trying to wait on Megan. Were those little microscopic cameras supposed to arrive in my office today?
Yeah, that's what Amazon said, but I never got a confirmation from them. I did look.
Okay.
Let me double check one more time. But
That's okay.
it shipped on Monday. So we'll see. Okay.
Yeah, for the teams, we have, one of you guys probably can describe that better, but we have a really cool little kind of magnifying little like camera that you can kind of plug into your phone, and you can magnify, like looking at leaves and insects and other things like that. And we were gonna gift that out to you all, just as something for you guys to kind of have. Not, you won't really need it for the competition, but it's just something in the future you guys and your teams and stuff can use. And then if you don't have, Gary, can you hold up that little guide?
Sure.
I know Cornhusker Kids, you probably have that guide, and I'm sure Aaron, your team have that. Brooke, maybe if you don't have it right away, maybe the extension office could get you some. Otherwise, like I said, I'll get them in the mail tomorrow, and they'll put kind of an express rush on it. But
okay, thank you.
Yeah, you're welcome.
It's got everything you need in there. For sure. So would you go ahead, or?
Yep, go for it, Gary.
Okay. Well, I'm just going to show some weeds and talk a little bit about it, and then if you got some specific questions. But I know in the past, you can use pretty much anything you want as far as a reference, like that books in the Weeds in Nebraska or the Great Plains, and I know some guys, I think Aaron's team had a weed app or whatever. I know University of Missouri has a pretty good weed app, and some other ones too, but there's also some weeds in this one as well. And so it's good to know the main weeds that are causing trouble in crops. And then also the noxious weeds, it's important to know those as well, because they can be a problem. And Sarah Sivitz, who's an extension educator out in Dawson and Buffalo County, out in that area, she isn't able to with us tonight, but she's helping with this as well. So some of the things that's important to know, and you don't really have a whole lot of time. We're going to have, I guess, 20 minutes. Is that right, for each station to do things, but it's important to know like the growth cycle of the plant, if it's an annual, biennial, like, or a winter annual, or else a perennial comes back every year. Like some of the, bad weeds. Well, an example is Sericea lespedeza, which is more of a pasture weed, but it's a noxious weed in Nebraska now, and it perennial, it comes back every year and that. And then you can tell a lot by the leaf shape, the size of the leaf, how it's attached to the stem, and a petiole, that holds it between the stem and the leaf itself. And that's one of the big things, how you identify Palmer amaranth, that the petiole is always longer than the actual leaf itself in there, and that's always going to be true. And then the stem, there's different, like a round stem is more normal than the square, like the mint family. And then like a sedge, they're the triangle shaped one, like yellow nut sedge, an example. And then a lot of time flowers, the shape of the flowers, the color and arrangement, if you have maybe 5 petals or whatever, they're going to be a big thing you can identify. And then fruits and seeds, and then how they spread, like velvet leaf, how those seeds are, and some of the tick seed and a fruit example is like pokeweed, gets that purple fruit on it. And then also it's important to know if there's something that's poisonous. Like an example is, I've seen a lot of this around Nebraska in the last few years, is poison hemlock, and we've actually had some animals in southeast Nebraska that were poisoned, and actually a couple people got sick as well. So there's seem to be more and more of that all the time showing up in the ditches, unfortunately. And then the root type is a big thing to know. Rhizomes, which travel underground, and one plant I know we've had it in the past, and it's shown up more at least in southeast Nebraska, is Johnson grass. And if you dig up, it kind of looks a lot like kind of like sorghum or 4-H sorghum, that type of thing. You dig it up, it's got these big roots, rhizomes on there, how it spreads. And stolons, basically above the ground, kind of rhizomes that are on top of the surface. And then just a straight tap root, or some plants have fibrous roots. These are just some things that it's good to know, that you can help identify some different weeds. Like I mentioned, the Weeds of the Great Plains is an excellent resource. A lot of, pretty much everything. We're only going to have 10 weeds, but we'll have like 5 questions, but they're pretty simple questions in that 20 minutes. And if you guys want to, some of the common weeds that we showing up that we'll just have pictures of it in there. But then like examples of sunflower, just an annual common sunflower, which is an annual plant, and it can cause definite problems. Anybody know what this is? Interesting, you see that bug up there in the corner, praying mantis up there. And this is giant ragweed, but and I know some places, down in our area, we have some that is resistant to glyphosate. So it's has problems with that. But it emerges real early in the year. So it's good to, if you have a field that's got a problem with it, you may want to try to plant that field later and get it all emerging, so you can kill it before that comes up. But it's an early emerger. And it's just probably called a winter annual, or foxtail barley. And it's usually not a big problem, but a lot of times it goes, and you'll see it going in pasture or wet areas as well. Like there's foxtail barley, and there's one called little barley. We usually don't have those on the contest because they're spring, but since we're just doing pictures, we may have some like that. Some like downy brome, and some of those that are more kind of early spring type. And another one. And here, I hope you know this one. This is a major problem a lot of times, because it's resistant to especially glyphosate a lot, and what we call mare's tail. And it can act as either a winter annual, but sometimes it can come up early in the spring, or just a summer annual, but it can be a problem. And we have found a lot of people are using cereal rye as a cover crop, but it seemed to work pretty good in suppressing mare's tail. And that's one thing that cover crops are shown to help out with that. And this weed's been around for a lot of years. I know since I was a little kid, I had to pull it, cut these weeds. It's a noxious weed. It's got a pretty flower, but it musk thistle in there. And that's something, noxious weed, if it's listed as a noxious, there's usually a weed inspector in your county, and like if you have puts pasture land, you have to watch out for that and get those out of there, or else they may, if you don't take care of them, they may do it for you, but then they'll charge you for it. Regard to that, but it's been around a long time, and actually I think it came from Europe initially, but I worked in Missouri a while. They have a thistle weevils, and actually I think it came from Nebraska. These thistle weevils get in the head and kind of help control it. That's a biological control. And then there's a rosette weevil too. And then like a musk thistle is like a biennial. The rosette will come up in the fall. That's the best time to kill it, in the rosette stage. And then it'll come back in the spring and kill it before it starts bolting, because it's a lot harder to kill then. What happened there? Oh, I don't know what that's about. We mentioned this earlier. Anybody know what that is? It's one that we call pokeweed. It's usually not a big problem, but a lot of times it, and then I think which is kind of ironic, I think maybe the berries might be poisonous because they always, I think you can eat the leaves. I know you guys heard that song, Poke. In southern part of the United States, they eat it, I think. Poke salad, Annie. But it grows a lot of times in wet areas and that, and I actually do think the berries are poisonous in that, but it's easy to identify.
I do get questions about that at the office almost every year, Gary. So
You do? Yeah, well, can I eat that? I see those.
yep,
berries.
they want to make it for jam, and I'm like, nope.
Or the seeds, how they move when animals will catch on animals, and very common. Well, a lot of people call it buttonweed, but velvet leaf's a common name. But I think the seeds of velvet leaf can stay in the ground like 30 years, a long time anyway. Something's causing weird that I'm having a problem advancing my slides.
Are you on like Google Slides or something, or?
No, I don't understand why.
Fun fact about velvet leaf in the meantime, it's originated in India.
It did.
In the
Tom
soft. Go ahead.
Tom Hogemeyer was telling the story that it was like either a Secretary of Ag or a senator's wife that liked it and then brought it back.
Yeah.
Because she liked the velvety leaves. Not sure how she didn't tolerate the smell, but.
So it's crazy with these weeds. It seems like somebody thinks they're pretty, and then we end up with the mess later on.
This is lamb's quarters, and there's a lot of it around for sure. Actually, I think you can eat it as well in regard to that, but it's a tough one in springtime. And then field bindweed, another one. And the one way you can tell, and I was just looking at some last night, about underneath if you pull up the flower, underneath the flower about 1.5 inches, there are 2 bracts on each side of the stem. They show up there, bracts there that they show up. So that's one thing with field bindweed. And this one is kochia. It's not so much a problem in row crop land on waste areas, but out in west where there's a lot of wheat grown, it's always been we call it fire bush is kind of a name farmers call it because it'll get 6 foot tall and kind of reddish and actually, I think the cattle like it when it's short like that, but you can't have issues with nitrate if they're grazing that as well, but it's a good one. Oops, sorry. And this is one of our main ones, water hemp, common water hemp. Go to that. And this is black nightshade. And those berries can be poisonous too. But a lot of times you get them and stain your soybeans, which isn't good. And buffalo burr, which a lot of times they got pretty sharp burrs on there, and a lot of times they'll grow in waste areas like where you're usually in a pasture where you feed hay or something like that. With where regular grass isn't growing well, a lot of times they'll kind of take over. And there's another noxious weed, Canada thistle. It can be hard to control. And cocklebur. And the difference, a lot of times when they're small, cocklebur and sunflowers look quite similar, but the cocklebur has the spots on the stem. And I think when they're really young, they're poisonous to pigs, like when they're really small, cockleburs are. Another common weed that's around, not usually a big problem in waste area, is witchgrass. And you see it's got the hair, it's really hairy. There's that shows up, and it's a way to identify it because it's really hairy. And here's another noxious weed that usually grows in around the rivers and stuff, is Phragmites, or I guess or reed, common reed. But it's kind of ironic. It's spread and it's all over. This was actually in my brother's pasture, and it's not around a river or anything. It's a little wet area, but it's way above any creek, a couple miles away from anything. But I'm assuming they just spread by probably maybe waterfowl, or there's a lot of, and the seed spreads a lot in the wind. But it's Phragmites is another name for it, but it's a noxious weed as well. And here's another one that's been around for a long time, leafy spurge. It's a problem in pastures, and the spurge has got that milky substance. All the spurges have that. And I mentioned this earlier. A lot of you may not have seen this. It's called Shirishia lespedeza. If you ever go down or drive around in the Ozarks down there, there's a lot of it down there along the roads. Missouri don't do a very good job of controlling it. And it's really bad in Kansas, but unfortunately, Nebraska, they planted it as deer feed in a lot of their state parks initially, and that wasn't a good thing. So, but now it's kind of like got 3 club-shaped leaves right on the stem, and it spreads, prolific seed producer in there. And this is probably the worst weed in Nebraska, in the United States, probably a Palmer amaranth. And there you see the petiole is longer, and it doesn't hold, but that's one thing. It can't have these chevrons or watermarks, whatever you want to call them. that has that. They don't have to have that though, but you can see that the petiole is just as long as a leaf. Oops, sorry. And this one up here is purple loose strife. Sorry, that's another noxious weed. It goes in wet areas in there. And of course, they used to be, sometimes they would sell it. Earl May used to sell it as a ornamental, but in wet areas, he goes can really take over. They used to have some at NREC up there in the corner of the area there when I worked there several years ago, and it was pretty much took it over. And when they realized it was a noxious weed, so then they controlled it. And this is one that a real pretty plant, scattering rush, or equisetums, another term, or horseweed, or I should say horsetail. I'm sorry, mare's tail, horsetail. And it's got, kind of looks like little bamboos in there, but it's really tough to kill. And pretty much any chemical, if you use on it, Telr, that's the one that works on it, but you're not gonna be able to plant much corner beans there for a while. And then this one that's kind of showing up some is called common teasel there. It's got the purple, then there's also cutthroat teasel. I mentioned Johnson grass earlier, so that looks like. And your last one is cutleaf teasel. There's a little bit of it coming into the state. There's not a lot of it. Some of the states to the Missouri and Kansas, there's a lot of it, and you get farther east. So I was going to ask, oh yeah, I mentioned there'll be like 5 questions, but a couple of, well, one of the questions, this is one that, well, when something similar to this, I said like, mare's tail can emerge in the fall and act as a winter annual. Is that true or false? Well, that's true. I mean, it can, and sometimes it can emerge in the spring, but it can do that, and so that's true. And I went over these. Serratia lespedeza and leafy spurge are noxious weeds, and that's true as well. So we'll have, I mean, the questions will be pretty simple, and maybe multiple choice, but it won't nothing be too difficult. So that's kind of what it'll be like. It'll be, I mean, when you have the weeds there and people, it's a lot easier to look at them when they're there in person, but it's just going to be pictures. It'll be, we'll try to make it so you can identify them, and hope if you have some books, it shouldn't be too tough. So. Megan, want to add anything? Or any other guys?
I think you covered it pretty good, Gary.
So do you guys have any questions about the weeds or the weed portion since Gary's on here? Yes, if you think of something, feel free to type them in the chat box or, reach out with an email. Otherwise, who's up next here?
It's me.
Megan? Okay.
Okay, so we're gonna do the corn growth staging, and I sent it to Gary, so sorry, Gary, if you don't want to flip through it, you don't have to, but go ahead and go to the one. Yeah, perfect. Okay, so one of the big things, especially in Nebraska, and we've had a lot of pop-up weather this year, is that we've been having a lot of questions, obviously, about some hail damage early, and so this year, obviously, it's a lot easier when we're in the field to have you go out, split the stalk, count the collars. And so that would be the most agronomic way to do it. So that's one way to grow stage. But then when it comes to hail, the way that a hail adjuster would treat that is a little bit different. And they do the counting leaves method. And that counting leaves method is just when 50% of the exposed leaves are counted. And so the collar doesn't necessarily have to be showing, but the leaf will have to have that decided droop for then that to be counted. And so we usually don't talk about that too much at the crop scouting day or at the contest, but that's something to keep in mind. And then the plant height. And so the one we're really going to talk the most about is going to definitely be the counting collars, because that's the true agronomic and the true growth stage of the crop. And so the counting colors one is the one we're going to use, but the counting leaves may be something that may be a question that we ask you about. So, you know, there may be one where we ask you to grow stage it vegetatively, so the counting colors way, and then one where we ask you to do it counting the leaves to compare, you know, what your results are, to see, you know, kind of how that changes how you, I guess, look at the crop, and then also how you decide where it is in its vegetative state. So go to the next one. And so with corn, remember that if we're doing the counting collar method, and this is also in the pocket guide when you get it. So I would highly recommend using the pocket guide when you are going through the growth staging portion, because sometimes you gotta check yourself. I do it too. But for the vegetative stages, remember that it goes from VE all the way up until it's V tassel. And remember that V tassel is obviously when the tassel's showing. And so that's when we end the vegetative stages and we become reproductive. So then you would start naming them in the R's, or the reproductive. And so again, this is all in that pocket guide. It's just a good reminder. And the other thing I wanted to mention is when we transition from V6 past, so once we're right around V5 to V6, that's when you need to start not only counting the collars, but you're going to have to start pulling the plant up and splitting the stalk. And so you can go to the next one, Gary. So again, when we do the collar method, when we're counting the collars, we are looking at all of the visible collars, including that first leaf or the seed leaf. So we're going to count up from there. And remember that once we get to the V6, that transition from V5 to V6, rapid growth occurs then. So that's when you're going to have to pull the stalk up and split the stalk. And so we are going to do one more where we're just looking at the vegetative stages. So if you can click to the next one, Gary. We skipped one, sorry. Okay, for example, so these are two that I've been looking at. These are two from last year. And so you can see that we have one, that was that first leaf. We have two, the collar showing. Three, we have the collar showing. Four, we have the collar showing, and five. So we'd count it at V5. So then if we put one more leaf collar on, then we have to start pulling it up, splitting the stalk. Always cut the stalk away from you. You don't want to stab yourself like me. That's not a great time to be alive. So don't do that. And then if we move over to the next one, you can see that we had the first true leaf at the base here. I didn't do a very good job taking the picture, sorry. The next, and so this is going to be first true leaf, collar showing, collar, so we're at V4. So it's really, really, really important to make sure that you get that first one when you're counting collars. And so once it gets too big, we start losing leaves. So that's why at V6, we have to transition into pulling it, splitting it. So when we split the stalk, so that's the next picture. So when we split the stalk, it gets really, really important to look for those stacked nodes. Those are in the red triangle there. And so in the red triangle, you can see nodes 1 through 4, and then 5, 6, 7, and 8. So it's really important when you split that stalk to find those stacked nodes and then count up. And another thing to do when you are taking the growth stage of something that is past V6 is count the collars that you can visibly see first, then split the stalk, find 1 through 4, and then count up. And that's just a good way to kind of gut check yourself. So if you're having trouble doing it the first couple of times when you split it, if you split the stalk and you use food coloring, like just really cheap food coloring and water, you can stain the nodes. And so you'll have a good conglomeration of that color right where the nodes are. And so this is how we used to, teach people when we first started. So if you're really struggling, just go get some like blue or red food coloring, slap it on the inside of there, and it'll make it a lot easier for you to see those nodes very clearly. And we can go to the next one, Gary. And so again, just so you can see, those stacked nodes do get really hard to see at times, the 1 through 4. And so you can see we have 4, 5, 6, 7, 8, 9, and 10. And so again, before it reaches that V tassel, so VT, you're still counting those, you're still counting the collars, but once it's past V6, you have to start pulling the plant up and splitting the stalk to get the true growth stage, because you can be off by 1 or 2 leaves, which could make a big difference in your management. So that's just something to think about when you're doing the corn growth stage and development. And Gary gave you some example questions, but really from my perspective, the big thing will be for the corn growth and development is just being able to grow stage a vegetative plant, and then being able to grow stage in the reproductive. And then from there, it's going to be a lot more just on ear development or environmental factors or hail, things like that. So those are just some things to keep in the back of your mind. Please use that pocket guide because it has a lot of information about anything I would ask you. So yeah, so that's all I had. So if there's questions, go for it.
You got some good pictures there, Megan. It's nice.
Thanks. Jenny shared some. So this one is Jenny's, and the one before was from pesticide. The other thing is when you are taking the stand counts, if you do take a stand count, make sure that you check the width of the rows. Last year, that was something we not really struggled with, but just make sure that you check that you are in 30-inch rows for corn. When you do that, remember it's 17 feet and 5 inches. So just check yourself. And then for 20s, if it would happen to be on 20s, remember that you're looking at, and I gotta double check myself on this real quick. But if you're on 20s, because 20s are a lot more popular now, it's 26 feet 2 inches. So that was all I had.
So what's the answer to the question on that slide? If the pesticide is labeled JNS, is it safe at this stage to add it?
So for this one, it was not safe because it was passed. So it was labeled up into V8, I think. And so this one was V10, and it was past the point where it was safe to use the product. Because it caused some ear formation damage. But those are things where, like, that's probably a little bit too hard. So we probably wouldn't ask that unless you had the label in front of you. So that was a good question. Anybody else? Okay, that's all I got. Nathan and Michael, you're taking her over.
Oh, Nathan, you're muted.
Sorry, smaller screen. Thank you. So it's really good to follow Megan. It's very similar for soybeans as for corn. Actually, it's a little bit easier. There is a pretty common growth staging system that's used universally for soybeans, actually developed by Fair and Cavanis. And so we have both kind of two major groupings. We have vegetative growth stages, which we'll go through briefly, and then reproductive growth stages. So very similar to corn, where in vegetative growth stages, the abbreviation, we just use the letter V instead of vegetative. And same thing when we get into the shortened acronym for reproductive is just the capital R. So for soybeans, the V stage or vegetative stage, in a way continues on in the background through flowering. And so we'll talk a little bit about that in terms of nodes. To start off with, this is again the 2019 pocket guide. So if you don't have a copy of this, like Brandy said, she'll be sending you one. First stage is VE, and E in this case is emergence. And so that's fairly standard if you see cotyledons pushing out of the ground. And in terms of emergence, it's a little bit different than corn in the sense that the growing point is already above ground when those soybeans push out. In this case, what you see are actually is that soybean seed pushed out, and so that hypocotyl. Those cotyledons are an energy source for the soybeans early on, both for nitrogen and other nutrients. And so we can talk a little bit about when we get to V2, V3. When we move on, I wanted to go back. For staging, we have VE, emergence, and then we have VO, which a lot of times you'll see VC, which means cotyledons. And then we have V1, which is the unifoliates. And then when we get, and that's when this first starts here at V2, is when we have the first trifoliate is out and the second pair of trifoliate leaves are no longer touching. So I'll show you that in a picture. And so you can get off one real easy because you just count it. Don't think about maybe that last set of trifoliate leaves because they're fairly small. So for example, here's V1. We have our first two set of cotyledons, and we have two growing points at each of those cotyledonary nodes or axils. And then we have our unifoliate leaves, so our first leaves, and those are opposite of each other, and so are the cotyledons. So when you look at a stem, and if you look at leaf scars, because these cotyledons or unifoliates can fall off, they'll be opposite of each other. Same thing with IDing weed species, like Gary was going over. You can have different leaf arrangements, opposite, alternate, world. So these first sets are opposite. And then from the trifoliate on, they alternate across the stem back and forth. So they're not directly across from each other. So this is V1, the first trifoliate, the leaflet's edge are no longer touching. So once those expand enough where you can see each individual leaflet of that trifoliate. And there's some argument over, it'll say here in the book, it's trifoliate. That is that first set of three leaves. So example, here's a trifoliate. It's actually trifoliolate, if you really want to get technical. And so we actually, if you look, it's in the book or the guide, you might see both spellings, but it's a trifoliolate, which is the three leaflets, the rachis, peduncle, and then essentially the petiole. So you have some different parts of that overall leaf. So just like an ash tree or a walnut tree, even though one leaf actually includes all those leaflets. If you look at an ash tree or a walnut tree, same thing with soybeans. So here's a really good example in this picture. We have our cotyledons, then we have our unifoliate leaves, then we have our first fully expanded trifoliate, and then this last trifoliate, you can see each of the three leaflets of that trifoliolate. And so in this case, this is now goes from V1 to V2 once these, you can visibly see that they're not touching anymore. So this is a V2 plant. So if these were tightly packed and still close together and you couldn't see the three leaflets, then it would still be a V1 plant. One thing to note on growth staging, whether it's corn or soybeans, rarely is the whole field and every plant in that field the same growth stage, right? So things emerge at different rates, whether it's due to soil temperature variability, depth in planting, for numerous reasons. Soybeans are the same thing. Whenever you stage, it's based on 50% of the plants in that field or 50% of the plants in that area are at that growth stage or above. So if you had 6 plants that looked like this and you had 4 plants where these 3 were still tightly packed and you couldn't see them, it would be at V2 because six out of the 10 were at V2. So whenever we've had it in the field days in the past, that's where we've gotten some teams will stage it wrong because they might take the most advanced plant. One plant was V2. All the other 9 plants, if you looked close, it was really hard to find another V2. Everything else was V1. In that case, the growth stage for that field or plot would still be V1. So remember, the 50% of the plants need to be at that stage to move to the next stage. So we keep moving through trifoliate stages here. At V2, back here, we're still relying on the cotyledons, and then we kind of move over at V3 where we're soil nutrient uptake from the soil becomes pretty critical. We can see in some cases where early soil nitrate, nitrogen availability, nitrogen will cause growth differences early in soybeans. And so we just keep going through those vegetative stages, okay? So how many nodes can there be, or VN or nodes can there be on the plant? It does depend on the year and the variety. It's not uncommon to have 21, 22 nodes at the end of the year. Does anyone know from V1 to R5, what's the rate that each one of these trifoliates or nodes appear? How many days in soybeans does it take, roughly from V1 to R5? Any guesses on days? Cornhusker kids? So if you can remember 4, that's the round. It's really close to 3.7. And when it comes to dicamba damage or drift damage or herbicide damage, if you can see when you first started cup leaves and what trifoliate that was, you can kind of figure out when the herbicide drift could have occurred. So that's something to look at when you have a bigger plant. Let's move on to reproductive stages real quick. So we keep moving through reproductive stages until we start seeing flowers. And as soon as we start seeing flowers, as soon as 50% of the soybean plants in a field have 1 flower on it, we are then at beginning bloom, which is the first reproductive stage, okay? So growth staging, so you could have a plant that's V5, and then right next to it is also a V5 plant that has 1 flower, okay? So in that case, you'd need 6 out of 10 to have a flower on it to move from V5 to R1. That's not a set time period. It might take till V7, for example, and then at V7, you start seeing the things switch over to flowers. And we won't get into the reasons in this contest as to why. Whenever I think of reproductive stages in soybeans, I really think of 4 main reproductive stages. I think of them as pairs. So the first set is your beginning and full bloom. So that's pair 1. The second pair is beginning and full pods. That's pair 2. Pair 3 is beginning and full seed. And the last one is a beginning and full maturity. So you can see we have the same beginning and full for each one of those four stages of reproductive growth. One thing that's a little tricky, I think is always good to remember, when we go from bloom, R2, into pods, we change what the number of nodes that we're looking at in terms of determining growth stage, and that really throws people off. So for R2, you have an open flower at one of the top 2 nodes with leaves that are fully expanded and out, okay? All of a sudden, when we get to beginning pod, now we're looking at the top 4 nodes. And so a lot of people will see 4 because it's 4, 4, 4 for all of these stages, but R2. So R2 is really the exception, okay? So just remember there is an exception to looking at the top 2 nodes at that stage for flowers versus for pods and seeds. We're looking at the top 4 with a fully expanded leaf. So let's look at a few. Here's an example of the flower. It's anywhere on the plant. And then we move into R1 as long as 50% of the plants have flowers, a flower anywhere. R2, we're looking for a flower out on one of the top 2 uppermost nodes with a fully developed leaf or trifoliolate. So here we have a flower here. This one right here, for example, is not fully expanded yet, right? It hasn't finished growing or completing. So we just need to have a flower at one of these 2 here, and we do, okay? The reason this matters on flowering is a lot of herbicides, when you can, kind of the end in which you can apply those herbicides change based on these reproductive stages. For some herbicides, you can apply to R2. In some cases, it's beginning bloom or R1. So that beginning to full bloom and into pod set, it really does matter on growth stages when it comes to when we can apply herbicides according to their label. So that growth staging, similar to what Megan went over with the non-ionic surfactants, growth staging becomes pretty important part of understanding for herbicide management or weed management plan. Then once we get into pod set, we have the beginning and full pod, beginning and full seed, beginning maturity, full maturity. Again, this book is really helpful because there's certain lengths that we're looking for pods, certain size we're looking for seeds. I'm not going to go through those, but I encourage you to review those before the contest. I guess there's a lot more there, but that's kind of the basics on soybean growth staging. I would expect to be able to know how to determine what the growth stage is, both for a plant, a picture we show you at the vegetative stage, as well as the reproductive stage. And we'll make the pictures, we'll have pretty good pictures. Since we're not in the field, we have pretty good pictures where it'll be fairly clear, I think, based on the picture, as long as you can do this, what growth stage it is at. Any questions on soybean development and growth? We got one group left. Okay, that's all I had.
So let's see here. That everybody has went, correct?
I didn't know if Michael has anything. I'll stop sharing.
I don't have
Michael,
anything. I don't have anything different. I'll just give a hint out that Nathan did cover, no matter how little he covered it, he did cover everything that we talked about for our competition in his talk.
And I put the NIS information, it's from Purdue. I put that in the chat in case anybody wanted to look at it, because that actually is a really good question. So might be something to think about. Maybe not that specifically, but something to think about.
Great. That sounds good. Well, are there any questions from team members kind of about the content or questions about the contest itself, like how we're going to kind of do this? It's basically, and I'm going to do some more practicing with my staff and stuff so I get the hang of it, but we'll basically have you guys put you into breakout rooms. So I like how at Cornhusker Kids, you guys put the name of your team there. So if you guys could kind of do that, that just kind of helps me keep track of who's who. And I would basically just put Cornhusker Kids into whatever breakout or rotation that they're going on. So if they were going to be going to work with Megan's session, I would put you in Megan's room, and you'll have 20 minutes in each room to to answer those questions and go through everything. And then we're going to have kind of a 5-minute break just to make sure that I don't mess up, or that, I don't have bad connectivity issues or anything like that. So we'll have kind of that kind of little leeway time, or, if you need to use a restroom, get a drink, some kind of stuff like that. So that's kind of how this basically is going to work. And, if it works, and if it's safe and you practice all the directive health measures and all that stuff, ideally it would be good for each team to kind of be together. So Cornhusker Kids, I know you guys have 2 teams, so if there's 2 locations, where team 1 can kind of be, and then team 2 can kind of be at another location. That'll just help the teams be able to kind of answer, or talk amongst each other and try to figure things out. The kind of test part that we've had in the past, we're going to do a fun Kahoot type of game. And so some of you maybe done that in school. I know my daughter loves it and has done it a lot. So there'll just be 20 questions, and new this year, the teams will be able to work together as a team. So in the past, you've done the tests individually, but this time, before you blurt out an answer, I want to make sure we say, or that you guys, whoever's trigger happy, because you'll be needing to use your devices. So you can either use, your laptop or your phone. We tried it with amongst the judges and stuff, and I think everyone pretty much used their laptops and stuff like that. So that's just kind of the real quick part of it. You should have all received the schedule today and the letter with kind of more of those details and other pieces. But are there any other questions you have, or judges, anything I'm leaving out that we talked about yesterday?